Hey guys, this is Mr. Wilhelm here again. I'm going to do an example of a dihybrid cross Punnett square. This matches your worksheet that was given to you in class 10.2, Part 2, dihybrid cross practice, practice number 1. So let's read the problem here. What are the phenotypic ratios that result when an organism that is homozygous recessive for two traits is crossed with an organism that is heterozygous for two traits? So the first thing I'm going to do is highlight the fact that we have one parent that is homozygous recessive for two traits, and we have another parent that is heterozygous for two traits. So what we need to do first is write the genotypes for those two parents. Okay? And how that's going to look is something like this. Homozygous recessive for two traits is going to be little a, and then little b. Okay? Now, I'm getting those genes from this key right here, which tells me that capital A is long arms, lowercase a is short arms, dominant B, capital B is black hair, and lowercase b is blonde hair. Okay, so the first genotype for the first parent is little a, little b. Okay, then we're going to cross that with a heterozygous for two traits. Heterozygous for two traits is going to look like this. Big A, that's heterozygous, and big B, little b, that's also heterozygous. Okay, so now we have the parent genotypes. Let's just call this one male, and we'll call this other one female. Okay? Now what we want to figure out is the sex cells or gametes that these two individuals can make. And the way we do this is use a method called the FOIL method. With FOIL, it's just like algebra. The FOIL method, the F stands for first, the O stands for outer, and the I stands for inner, and the L stands for last. So how can we apply that concept to figuring out sex cells? Well, let's do the male sex cells first. The first letters we're going to focus on are the first letter for each trait. That's A and B. So the first sex cell is just that, A, capital B. The outers are going to be, I'm going to put a black circle around the outers. There's one outer, and here's the other outer. And so those two combined give us capital A, lowercase b. Now we're going to do the inners. I'm going to put a rectangle around the inners. That would be these two, which is lowercase a, capital B. And then we're going to do the last, and the last in this case are the last for each trait, which is a little a and a little b. Little a, little b. Since these are sperm cells, let's just put a little circle around it with a little tail to indicate that they are actually sperm. Now what have we done here? We've actually just completed all of the possible combinations that these letters can make with these letters. Okay, and there's 4 different combinations of sex cells or gametes that can be made. Now we're going to do the same thing for the female sex cells. The first are going to be little a, little b. The outers are going to be also little a, little b. The inners are going to be little a, little b. And the lasts are going to be little a, little b. I accidentally did that one in green. It doesn't really matter. They should all be red, but these are all going to now represent the female egg cells. I'm just going to put a circle around each one. Okay. So these are all eggs, and these are all sperm. What we are using a Punnett square for is to determine all of the possible combinations that these sperm here can make with these eggs over here. That's why Punnett squares are helpful. So we're going to go ahead and rewrite then the sperm sex cells on top. Big A, big B, big A, little b, big B, little a, little b. And we're going to rewrite the female egg cells on the left. Little a, little b, little a, little b, little a, little b, little a, little b. Okay? So now what we're doing is just like any Punnett square, we are looking for the combinations of these sex cells. So basically what's going to happen when this sperm cell fertilizes this egg cell? And we're going to write that combination in the box. So I'm going to use one color now. I'll use a new color. We'll make it black to represent the new individual. And we always write the dominant gene first for each trait. So we have big A, big B, little b. And really, it's going to be the same combination all the way down this column. Big A, big B, little b, all the way down. Okay, so then we come over, we do the next one. We want to find out the combination of this sex cell with this sex cell. And what do we get? Big A, little b, little b. And they are the same all the way down. And then we go over to the third column. We've got little a, big B, little b. Little a, big B, little b. Little a, big B, little b, and again. Okay? And finally, the last one. Little a, little b, little b. These would all be called homozygous recessive for both traits. Okay? So, now we've completed all of the possible genotypes that could exist from this cross, and we're going to figure out the genotypic and phenotypic ratios. Well, there's really going to be, in this particular case, there's going to be 4 different genotypes, and they're listed in this top row. So if we write them out here, we get big A, big B, little b, and we want to find out how many times that occurs out of 16. Well, let's highlight them. Big A, big B, little b is also heterozygous for both traits. So that's heterozygous for both traits, heterozygous for both traits, heterozygous for both traits, and heterozygous for both traits. So that comes out to 4 out of 16, which is also 25%. That's supposed to be a semicolon. Okay, so what's the other genotype that we get? We get big A, little b, little b. And let's highlight that in a different color. So that's heterozygous for the first trait and homozygous recessive for the second trait. That's all of these. So we get 4 out of 16, semicolon, or 25%. And let's do little a, big B, little b. Well, as you can see, that's all of the results that are here, and that's going to be equal to, again, 4 out of 16. I think you can probably see where this is going. That's 25%. And let's color all of those green. And then last but not least, this last column is homozygous recessive for both traits, little a, little b, and that's equal to 4 out of 16, which is 25%. And let's color those purple. So, that's the genotypic ratios. Remember, a genotype is the letters that represent the genes, okay? So we know we're talking about genotypes when we see a bunch of letters together, and they occur in pairs, okay? Unless you're talking about sex cells. The sex cells have just one letter that represent each trait. Phenotypes, on the other hand, are what you see as a result of a particular genotype. It's the observable characteristic that is the effect of a genotype. So there's going to be 4 possibilities here, and now we go back to our key to kind of figure this out. We know that the dominant A is long arms and the recessive is short arms. So this first one here is going to be long arms and black hair. I know that it's long because of that A, and I know that it's black because of that B. What about the second one? This one here is also going to be long, but this one's going to be blonde hair because it's got both recessive alleles there. This one will be short and black, and this one will be short and blonde. So now we have to go back and see how many times those occur, and really what we're going to find out is that they really match up with the genotypic ratios on this particular problem. That won't always be the case, but in this case it is. All of these that are highlighted in yellow are going to be long and black. Okay, that's 25%. All of these that are highlighted in sort of this teal color are going to be long and blonde, 25%. All of these in green are short and black, that's 25%. And then all of these in purple are short and blonde, 25%. So that is the answer to the first practice problem on your worksheet. I hope that was helpful. I'll see you in class. Thank you. Bye.